Given the amount of resources spent creating new drugs, you might expect significant discoveries to happen all the time. But the reality is that drug discovery is burdened by significant challenges. Why is that? To start, just finding the molecules that could be candidates for new drugs is slow and tedious. A protein is first hypothesized as being associated with a disease, then a drug is engineered to bind to that protein or target to treat the disease in some way. Both the hypothesis and drug must then be tested through clinical trials. These are a critical part of the process, but they're notoriously slow and expensive. Clinical trials fail about 90% of the time, with 40% of failures occurring in phase 3, the most advanced phase. At that point, the entire drug discovery process must be restarted. What leads to those failures, and what can be done to improve the chances for success? The most common causes of failure are either poor efficacy or problems with toxicity. Let's dive deeper into each of these challenges and their potential solutions. Efficacy failures happen when the original hypothesis was wrong and the target wasn't actually related to the disease, or when the drug molecule just doesn't bind properly to the target. These issues could be reduced if scientists had a better understanding of which target proteins are driving the disease, as well as how the candidate drug molecule might interact with those targets. Toxicity failures happen when candidate drug molecules have a negative effect that outweighs any positive outcomes. Think of these as very bad side effects. This typically happens when a drug interacts with an unintended target protein, or when it affects the intended target in an unexpected way. These side effects could be reduced if scientists had a better idea of how a candidate drug might interact with all the other proteins in a patient's cells in addition to the intended target. Modern computation provides a path forward addressing both efficacy and toxicity failures. There are techniques that blend AI and physics to make predictions about the binding between proteins and drugs even without detailed structural information. That speeds up drug discovery and allows scientists to set their sights on more complex targets that are related to disease but difficult to design drugs for. These techniques also allow scientists to get lab-accurate results for binding interactions from all possible human proteins, the entire human proteome, instead of just the target. This screening enables proteins with toxicity issues to be predicted in advance. By incorporating the vast data contained in academic literature, experiments, and other multidisciplinary sources into the drug discovery process, we can arrange all relevant information into a mathematical model known as a Causal AI graph. That graph connects potential causes to effects. Simulations are then used to check which cause and effect relationships really hold up, allowing scientists to select better targets, discover better drugs, and even structure better clinical trials for existing drugs. Together with proteome screening abilities, this accelerates not only the discovery of new drugs at the molecular level, but the entire scientific process, from target discovery to efficacy and toxicity prediction to clinical development and beyond.